Hmm. It was good. How was yours?
What?
I said mine was good. How was yours?
Oh, it was all right.
All right. Not long enough, probably.
No, it was pretty good length.
Good. Now you're ready to learn some biology.
Yeah.
Cool. Yeah, welcome back. Hopefully you had a nice spring break. We do still have 8 more weeks, so just a reminder not to lose, strength here. I do have an assignment for you, hopefully, and maybe not. It's possible you've seen this on the syllabus. This is usually a pretty fun activity. This is worth 30 points, so this is a good way to boost your grade if you do a good job. This is your infographic assignment. So I have this posted on Blackboard. This is due, oh my gosh, I'm blacking on the due date. It is due April 11th. So that is like 2.5 weeks or so. Realistically, I wouldn't expect that this would take you like a huge amount of time, but I do want you to be creative. I want you to especially be creative in your topic selection. So you'll notice a large majority of what I have outlined, and this is exactly what is on Blackboard. I have quite a few different notes about the topic. So the topic is your choice. It's similar to the research article report in that like you can choose whatever you want with a couple of caveats. I do have a list of prohibited topics. So you'll see these at the bottom. You cannot cover body systems, mitosis, meiosis, photosynthesis, cellular respiration, or organelles. For one reason, those are relatively boring. They're also fairly simple. So that's why I have those prohibitions. I want you to choose something that is a little bit more nuanced. So for example, maybe you would look at like poison acquisition strategies and poison dart frogs. Like that's something I would be interested in. And that's like a biology topic. You could look at how they eat specific types of ants, and then that's how they, acquire toxins. If you're interested in like health and human performance or like sports or anything like that, I mean, there's a lot of interesting things you could pursue. Maybe you look at like how group workout classes are related to like student performance or something. Like there's lots of different ways you can pick a topic that you're interested in and then find sources to kind of support that. The idea here is you're creating something that's like aesthetically pleasing, so it should be visually attractive. It should also be appropriate for the general public. So the more complicated a topic you choose, the more aware you have to be that like you need to choose words that are recognizable but get the point across. So that's kind of the vision here. You'll see I do have a couple of other requirements. I don't know why that word was leaving my brain. So you need 5 facts. That's honestly not that many facts. So 5 accurate facts. Appropriate for the general public. You need to have some sources. You can use lecture, but you do have to have 3 outside sources. That doesn't mean peer-reviewed. Peer review literature can be a good source, but there's other reputable sources like NOAA and government websites and things. They also all produce research as well. So reputable sources. For colors, you need photos. You need grammatically correct text. Those are the requirements here. So spend some time looking through this, thinking about your topic. If you have a question like, hey, Nora, is this a good topic? Just let me know. You can ask me in class. You can send me an email, whatever, and I'm happy to chat with you. If you're blanking on ideas, we can talk about things that you're interested in and potential ideas. So last thing, you do need to have a separate paragraph. 3 sentences is what I say, so that's very minimal, describing why this is important information for the general public to know, and then what strategic choices you made to effectively convey that information. So that's what your paragraph needs to outline. Again, very briefly, 3 sentences, 4 sentences, 5, that's plenty. Questions here? Cool. So I do have a couple of recommendations here. Adobe Suite or Canva, those are really good software systems for this. If you want to use PowerPoint, you're free to. But in general, I do recommend that you look up what an infographic is, like literally Google infographic if you're not familiar. They should be like 1 page and they should be like a vertical orientation. If you look back on the board, there's a couple of like infographic adjacent things where they're like kind of posters, but they're posters with information. Yeah, So you need to have a couple of pictures. So 2 graphics, I say that could be like a chart, that could be a graph, that could be a picture. And then beyond that, you need to have, for example, multiple colors. So if you had like blue text in one section, maybe you'd want like a pink triangle in the bottom corner and then like a yellow highlight or something. Does that make sense? Like multiple colors to kind of emphasize your point. Thematically, you can incorporate them as well. So maybe you use different like background colors or different text colors. Different shapes can be fun. to try to draw the audience in. Yeah, any other questions? And there are honestly quite a few good or moderately good, decent examples of infographics like in the hallway. So if you want to check those out, let me know. I can also send you examples if you want me to. So if you reach out, just let me know. I do have this set of reminders here for you. I did get your grades all up to date over spring break. So your research article reports are all graded, and I do have feedback for those on there. So if you're wondering why you got the grade that you did, I probably told you why in the comment box. So make sure you check that out. Exam 2 is going to be Friday, April 4th. That's a week from Friday. So I will post a study guide for you all today after class, sometime in the couple of hours after class. Infographic due April 11th. So this is kind of what we have going forward in the next couple weeks. As promised for the exam, I'll give you, I think I said a half piece of paper. So if you want to make notes on the half piece of paper, that's your deal. So like this size of paper, front and back would be cool. Questions on any of this? All right. We didn't have time to cover everything with cancer, so I did want to finish up some of the slides we were unable to get to. Importantly, we were discussing the cell cycle and the fact that cancer is essentially like a loss of cell cycle control. Now, a couple of really interesting features of cancer cells is the fact that they don't need normal signals to regulate the cell cycle. Now, this is an active area of research, and cancer researchers are basically looking to see, like, do cancer cells make their own growth factors or their own molecules to kind of push the cell cycle forward, or do they simply no longer need growth factors or those other molecular controls? So this is kind of crazy, honestly, the fact that they don't need those molecules to drive the cell cycle. That really explains why we see such rapid growth within these types of cells. Something else that's interesting, in culture, they do not stop dividing when those growth factors are depleted. So in culture basically means like in a petri dish, essentially you can like grow cells, and when the growth factors are depleted for normal cells, they would die. But with cancer cells, again, we don't see that happening. Now, most of the time, cancer is caused by genetic alterations, so changes in the genes that affect like important protein function. And something that I had also previously kind of talked about a little bit, cancer cells are able to evade signals for apoptosis. So does anyone remember what apoptosis means? Sure. Yeah, it's like cell suicide. It's like programmed cell suicide. So remember, most of the time, cells have the ability to recognize when something is going wrong, and they're going to flag themselves for death. They're going to mark themselves as problematic, and the immune system is going to basically find those tagged cells and destroy them. This is a really important feature. It's very good that our cells have this programmatic cell death because it keeps things like cancer at bay. It keeps other issues at bay. So cancer cells, again, don't have those types of restrictions. And all of these features basically underlie why cancer can be really challenging to treat. We also know that there's stuff we don't know. So this is, again, a really active area of research. And I think I've also mentioned, we do have a cancer biology class. So if you're interested in learning more about it, you can do that. Now, because of these features relating to like growth factors and cell growth, some folks refer to cancer cells as being immortal. Now, with a continuous supply of nutrients, cancer cells can grow indefinitely. This is kind of crazy. Now, something else that's really, really cool and honestly kind of interesting within like scientific and biology history, there is a specific line of cells called these HeLa cells. And if you went online right now, you could Google like HeLa cell vial. You could purchase them for a couple $100s. You could get a vial of cells. Now, these cells have been around since 1951, and they were originally taken from a Black woman who was under treatment for cervical cancer. So they took some cells from her cervix, and those are the cells that have been growing since 1951. So they've been growing for a very, very long time. And again, relating to this idea of immortal cancer cells, that is really interesting. Now, there's a whole book about this woman whose cells were cultured. She was not compensated for this. Her family never knew they took them. Her family was never compensated. And there was actually quite a bit of like ethical and moral issues associated with these cells. This is a common theme in biology and scientific research in general that we'll kind of continue to touch on. The idea that like sometimes really important scientific discoveries have been made, but they have had problematic ethical consequences. So It's good to kind of consider both sides of these things. I definitely recommend this book. You should read it. It's great. It's fascinating. And it's talking about the person whose cells they were taken from. So HeLa stands for Henrietta Lacks. Yeah. No, so she unfortunately passed of her cancer, and it was really honestly tragic. She was from a part of her city and she was not very well educated. I think she dropped out of school in like fifth grade, and her children, I think, also dropped out around high school age. So they also lacked education, and they honestly never knew, and they were profiting hugely. Like the companies were profiting off of these cells, like billions of dollars off of just her specific cells. Yep. I think they actually settled in 2023, so 2 years ago, because of this book, which is kind of cool. Yeah, good question. And I do have a note here. Normal healthy cells can typically only divide 20 to 50 times. That would happen within just, I mean, honestly, it depends on the cell type, but maybe a year, a couple of years. So the fact that these cells have been alive for that long is really outstanding. Any other questions here? Cool. Like I said, you should read this book. It's great. A little bit more about cancer and kind of this condition. So when abnormal cells, or if abnormal cells basically evade this programmatic cell death, if they evade the immune system, tumors can form. And the term tumor basically just refers to a mass of cells. And we could have benign tumors or malignant tumors. Now, benign tumors are basically these capsulated non-cancerous type of tumors. It's a growth of cells, but they're not problematic. They're not pathogenic. They're not likely to cause harm. A lot of times the treatment for a benign tumor, if it bothers you, would be just like basic removal. However, malignant tumors are problematic. They're cells who have genetic and cellular changes that spread to new tissues, and oftentimes they impair organ function. So if you know someone who's been afflicted with cancer, you might have heard this term malignant, or referring to like a metastases, which is like a spreading of malignant tumors through the body. Now, there are, of course, other types of cancer, like blood cancer, which is a different type of, basically like cancer cells are transmitting around the body via like lymph systems and blood systems. This is not necessarily referring to those cases, but again, there's a lot more detail we could go into. Cool. So just to kind of reiterate an important point here, a benign tumor is not cancer. We don't refer to it as cancer. Cancer specifically refers to cancerous cells that lack the ability for that control and that have potential to spread and harm an individual. So a malignant tumor would be considered cancer. Okay. Just to be clear with this figure, blue cells are representing like the tumor. And in this figure here, these blue cells are representing cancerous cells. You can also notice that unhealthy cells are associated with a different cellular shape that is characteristic as well. So if you're looking at visuals or illustrations of cancer, a lot of times we'll see these kind of like misshapen cells. Now, a lot of folks might already know what can cause cancer. Sometimes it's just heredity. Sometimes mutations that cause issues with like these proteins or these genes can just be something that you inherit from a parent or a grandparent. Oftentimes, viruses can actually cause cancer. So we know, for example, HPV causes cervical cancer. This is like a common link. There's other viruses that can actually cause cancer as well, which is kind of crazy. Viruses can basically affect the body, and then sometimes they can actually change the genetic material. So did you have a question, Dominic?
When it comes to like blood cancer,
Mhm.
is it malignant tumors that are traveling through your blood system, or is it just like small little?
It's more like cellular. It's not like chunks are floating around necessarily, but it's like cancerous cells would be circulating. But honestly, there are like different types. There's also like lymph system, and I'm pretty sure you can get cancer in that system as well. So that would probably be different. But to be honest, there's a lot of detail, and I'm not a cancer biologist. So why is it that sickle cell isn't considered cancer? Well, for starters, sickle cell doesn't like necessarily affect other parts of the body. So like a sickle cell disease can cause issues with like vision, and there could be other like associated impairments, but those cells aren't likely to cause problems in other cells necessarily. It's not going to like grow and spread. but basically it's just a classification. So those aren't cells that have like lost their control system. Those are just cells that have a different gene. Yeah, so sickle cell anemia is basically caused from a specific alteration in the protein. Yeah, good question. Anything else? I don't know why these are automatically changing. Yeah, so other things that can cause cancer, this you might have heard of, damage to your DNA in the form of like UV radiation, smoking, and chemicals. This is really common. There's a lot of instances throughout history where we can observe these types of examples. Certain types of like pesticides, herbicides, different chemicals that were used during wars in history have been linked to cancer. Other types of radiation, so if you think about like nuclear radiation, that can be a big problem. Something else, cancer can just occur randomly as cells divide. If we think about like folks who you know who have cancer, most of the time, at least typically, they tend to be older. And that's because over time, you would accumulate mutations, which basically means you're just more likely to like have more mutations, which could develop into cancer over time. So remember that all of these causes are basically associated with mutations of genes, which is basically what cancer is. So again, that's why we would see this usually more over time. And I was kind of just answering this question for you. Cancer risk increases with age. Why do you think that is? This data is a little bit out of date. I was looking for a more updated figure, but there's some kind of issues with sourcing public data right now in some ways, but essentially we can see here there's much lower cancer rates for folks who are under 50. Basically, we see a lot higher cancer rates as folks get older, again because of accumulations of mutations. We also see that there's accumulations of risk factors over time. So as you age, you're likely or more likely to have been exposed to harmful things, maybe secondhand smoke, maybe you've been in the sun a lot, things like that. Now, lastly, you definitely don't have to do this, but some students honestly like this. There's certain websites and self-quizzes, things, questionnaires and stuff that you can do to assess your own cancer risk, which some people like, some people find it kind of freaky, but this is an example from the American Cancer Society. It will go through and it'll assess your cancer risk based off of questions that you answer. So it asks you things like, does anyone in your family have cancer? And like, how old are you? And like, do you smoke? How often do you eat vegetables? Things like that. So if you're interested, you can do this. You definitely don't have to, but it can be sort of informative. Other things that I was kind of talking about previously for youths or for young people, the biggest cancer risks are like colorectal cancer and skin cancer is a really big problem for youth populations. Okay, we're going to go into meiosis. Come on now. Hopefully. Okay, so meiosis is one of our main topics for today. We're not going to probably get all the way through meiosis, and it's super important that you all feel comfortable with this material. So we will, I'm going to try to make sure we're going through at a reasonable pace. Here's a little comic. Mom says I can't do meiosis until I finish my interphase. Kind of silly. Interphase would be the cellular phase that would precede meiosis, is the joke here. Give me one sec. My clicker isn't working. Hmm. I don't know why this is happening. Okay, for today, I want to make sure that you all know the steps of mitosis, not mitosis, sorry, meiosis. I want you to be clear on distinguishing between mitosis and meiosis. This is something that honestly a lot of students struggle with. So it's really important that you're able to distinguish these two processes in your mind. And it's really important that you can compare and contrast them. I guarantee this will be like an exam question. If you go on to take literally any class in our department, this is something that folks will be expecting you to kind of understand and have a basic understanding of. Meiosis is critical for understanding genetics, microbiology, evolution. So this really is a very foundational topic. That's not to like scare you. It's just to kind of emphasize the fact that like these are really important processes. So really make sure it's clear in your mind. Definitely spend the time to kind of learn meiosis. We're also going to talk about how meiosis leads to genetic diversity. This is like one of the most important learning objectives of this whole course. Like genetic diversity is critical for the continuance of all species, maybe not all, but many species, particularly like humans. So this relates to that idea as well. Prior to going into meiosis, I just want to review mitosis really quickly. So without using your notes, and this might be a challenge if any of this information is like leached out of your brain over spring break, I want you to, on a scratch piece of paper, briefly draw interphase and the steps of mitosis. You're free to work with a partner. I also want you to include some of these important features. So centromeres, spindle fibers, chromosomes, and the nuclear membrane. I have some other labels I want you to include as well. So spend a couple of minutes here focusing on this. Like I said, try to do this without your notes at first. If you're like absolutely stumped, you and your partner, then you can look at your notes. But I do want you to try to do it without your notes first. And I do have some of these half sheets if you want one. There's not a ton, but they're left over from earlier. So you can grab one if you'd like. The last thing I want to make sure I'm pointing out when you're doing this illustration, something I want you to focus on, make sure that the correct number of chromosomes are shown throughout this process. So that can be a challenge, but make sure you're maintaining like the chromosome number that should be maintained. So yeah. Yeah, you're free to work with folks. Try to do it without your notes. If you need a little bit of paper, I have some. I can bring it to you. I'll deliver it. Do you want a piece? Ah, they're just languishing up here. I'll deliver you some paper. Oh my gosh. Classroom is so cursed with markers. Look at this. What is this supposed to do? Why is there never any good markers in here? Yeah, interphase is before mitosis. I'm hearing some talk about that. Yes, remember what happens in interphase? It's super important. Yeah, both those things. Absolutely. Yep, the cell grows and it duplicates its DNA. Yep, super important. Give you a couple more minutes here and then we'll keep going. How are we doing? Need more time? A little bit? Okay. We're going to go over this just a little bit. So I do have this visualization here. This is like a summary of the different stages of mitosis. So a couple of key things we're noticing here. I asked you to kind of focus on centromeres, spindle fibers, chromosomes, nuclear membrane. So when we're looking at this figure, like this dotted line would be the nuclear membrane. So a key feature there is it has to break down in order for the chromosomes to be able to be split. So that's why I asked you to include that component. Spindle fibers. Can anyone tell me something about spindle fibers? Where do those come from? What do they do? Yeah, they pull apart the chromatids, absolutely, the chromosomes. And they're generally like pieces of the cytoskeleton, right? Some other important features, you already talked a little bit about chromosomes. What about the kinetochores? This is kind of a challenge. What's going on with kinetochores? Any thoughts here? They make chromosomes. That is a good idea. I don't know that I fully agree with that, but it's a good thought. Say what? Something about them moving. So it is sort of related to them moving. Now, when we look at this figure here, the kinetochores are honestly hardly visible, but they would be like these little black dots. Now, kinetochores are basically protein structures, and they're the attachment site. So that's really key. They're the attachment site, essentially, for the centromere. So that's where they're going to have the spindles attaching, and that's where they're going to pull apart. So the reason that this is important, and I'll mention this again later, the mechanics of meiosis are really similar to the mechanics of mitosis in that the way chromosomes are essentially pulled apart is pretty much the same between these two processes. There are some key differences, but that's kind of why we're going to be reviewing here. So jumping into our exploration of genetics, there's a couple of key terms you need to know, and you might be familiar with these already. Genetics is the study of heredity and inherited variation. So heredity refers to like the transmission of traits from parent to offspring. And variation refers to genetic differences. So we can kind of visualize this idea when I say things like offspring are not identical to their parents. That's an example of genetic variation. Like if you have a sibling, for example, unless you're an identical twin, you probably don't look the exact same as your sibling. And that's an example of variation. But you probably look more closely related to your sibling, or maybe not. But you probably look more closely related to your sibling than you do like a random stranger on the street. That's an example of heredity. So these two ideas of heredity and variation, that makes up genetics. So genetics is a really broad kind of like category of biology. And importantly, hereditary traits or traits that can be inherited, that can be passed, are generally passed via genes. So that's how this process works. You've definitely heard the word genes before, and I'll go into a little bit more detail of review on the next slide. But up here I have the Jonas Brothers, not just for fun, although I do find it amusing. But this idea is essentially heredity explains why the Jonas Brothers look like their parents, which you can see in the bottom picture, and why they look more like their parents than they do pretty much anyone else in this room, for example. But then variation is why we see differences between the Jonas Brothers. Like we can tell them apart, right? Like they don't look exactly identical. So this is an example of these types of characteristics or these vocabulary terms. Questions here? I really went back into the archives for that bottom photo. So a reminder here, as I was just saying, hereditary traits are passed via genes. And hopefully you can remember that a gene is made up of DNA. And we've talked about this in lab. A gene is the basic unit of heredity. So the basic unit of how things are passed from parent to offspring. And genes are essentially like instructions. And when I say they're like instructions, I basically mean the specific sequence of DNA determines what amino acids are going to be combined together and what proteins are going to be formed. There's a very direct relationship between the genes present and the proteins formed. So if we see variation in the amino acid sequence, oftentimes we see variation in the protein function. A lot of times that can be caused from an issue within the DNA sequence. So there's a lot of kind of relationships here. Now, genes are found on specific locations in chromosomes, and I have a picture of that in a couple of slides here. But remember, we've talked about chromosomes before. They're essentially DNA protein complexes. So this is a beautiful example of a chromosome. Now remind me, how many DNA molecules are in one chromosome? One, did I hear? I heard a lot, and I think I heard one. Anything else? Any other thoughts? Okay, the correct answer is one. There's one DNA molecule in one chromosome. So this is kind of like a confusing way to think about it. But remember, DNA is essentially referring to the deoxyribonucleic acid, right? So it's just a macromolecule. It's a combination of nucleotides, right? That's what DNA is. And DNA is basically, you could chunk it up into different pieces, and those pieces could be genes. So genes are just bits of DNA, and we can have lots of different genes on one chromosome. Yeah. Correct. Yep. And basically the way that that DNA molecule is packaged, that's what we refer to as a chromosome. It's that like packaged up bit. So yeah, one chromosome has lots of different genes, but one DNA molecule, it has lots and lots of different nucleotides. So nucleotides, remember, are like these little subunits. And when we look at this figure, each of these little like colorful bands on this ladder looking thing, those are all representing nucleotides. So there's tons of different nucleotides. So yeah, this is where kind of the vocabulary starts to get a little bit confusing. So I do understand that this can be kind of hard. Now, in total, humans have something like 20 to 25,000 different genes. And I've mentioned this before. Remember, there's other organisms that have more genes. There's other organisms that have less. We also know humans have 46 chromosomes. Remember, we have 44 that are identical, and then we have one set of sex chromosomes. We're going to be focusing a little bit on those today. So the way genes are passed from parents to offspring are in the form of gametes. And gametes are what we refer to as germ cells. So if we talk about somatic cells, those are like autosomal, those are non-sex cells. And then we have germ cells, which are basically sex cells. So those are our two categories. Now you can kind of remember this word if we think, and I say these types of terms a lot because I think they're helpful when you look back at like the root word. So the word germ stems from the root word germen, which means seed. Sometimes that can be helpful. So gametes are sex cells, they're germ cells. And importantly, gametes only have half the number of chromosomes that somatic cells have. So that means they're haploid. They have one set of chromosomes. Whereas if you plucked a skin cell, we'd have two sets of chromosomes, right? They'd be diploid. And we've talked about this. So most cells in a human organism are diploid, except for gametes. Those are haploid. And the reason meiosis is so important is because this is what produces gametes. Meiosis is the cellular process that produces these haploid gametes, these haploid sex cells. Now the figure up here I'm showing are human gametes. So we have sperm and egg. And these are not to scale. Sperm is much, much smaller than an egg. Questions here? Now, when we refer to the process of sexual reproduction, this is basically the union of two gametes. So typically this would be sperm and egg, and we can see this process really simplified here. So we have this union of sperm and egg. This results in a zygote, and a zygote is a fertilized egg. So we can kind of follow the ploidy level here. We have a haploid and a haploid, and then they combine together. 1 + 1 = 2, so diploid is 2. So we have the haploids combining together to form a diploid zygote, which is that fertilized egg. And the process of sexual reproduction essentially refers to this joining of gametes. That's what this process is. And typically this requires gametes from two different parents. And what's really, really cool about sexual reproduction is that it produces offspring that have unique combinations of alleles. This is frankly a typo here. Unique combinations of genes is not what this should say. This should say unique combinations of alleles. I don't know why I wrote that here. But this is that idea of genetic diversity. We get differences because these gametes are joining together. This relates to that idea of variation. And I have a question down here for you. What would happen during fertilization if gametes were diploid? What would happen? Would it be a polyploid? Is that what you said? It would be four. Yeah, absolutely. So that's a great use of the word polyploid. Poly refers to like multiple, so we could refer to polyploidy as anything more than like two. So yeah, basically what would happen is if we had two diploid organisms, and by organisms I mean cells, if we had two diploid cells combining, we would have a tetraploid zygote, which would be really problematic because then if that fertilized egg went on to duplicate, all of those future cells following the process of mitosis would all also be tetraploids, right? They would have way too much genetic material. Now, most of the time, changes in ploidy level are fatal in vertebrates. So organisms with backbones usually are not capable of like living through this type of issue. Was that
Like strawberries, why is it that bacon?
Yeah, that's a great question. So strawberries are octoploids, which means they have 8 sets. There are some examples. So notably plants, we see this a lot. Plants are not vertebrates, so they definitely don't have a backbone, and they have a very different reproductive system that's frankly quite complicated. Things like fungi, yeast, which are small eukaryotic organisms, plants, they all have kind of different restraints. When we talk about meiosis and mitosis, we are mostly referring to like human organisms and other vertebrates. Why are they capable? Gosh, I honestly don't know that there's a super good reason for saying why. It's just it occurred and it doesn't cause problems. In general, we would expect like even numbers would be probably okay with the process of meiosis because we can split 8 into 4. So we could have two gametes that would form 8 again. But the issue would be with things like 5 or 3 or 7, right? Like you can't evenly split 7 in half. You can't have 3.5 gametes. Are there any that do that? I mean, there are like, mm, no, not that I'm aware of, but that doesn't mean they don't exist. Yeah, the issue of ploidy is really haunting. It's very interesting. And I'll talk more about it kind of as we move forward. But again, this idea, changes in ploidy level are typically fatal in vertebrates. There are some notable examples. This is another reason to love frogs. They oftentimes are actually able to withstand changes in ploidy level, even though they are vertebrates. So frogs have a backbone, but they can kind of do some crazy things. So that's why we should all love frogs. Okay, this figure is showing changes in the ploidy level throughout the human life cycle. So a couple of key features here. We can note the different colored arrows are associated with different ploidy levels. So again, haploid and diploid condition. We're also seeing some key features. So we can see the, I'm trying to think of the word, can't even remember, the structures where these gametes are produced. And then we can see meiosis is the process that basically takes a diploid and splits it into a haploid. We can also see here that fertilization process where those gametes are reunited and the diploid condition is restored. And as I was mentioning previously, the life cycle for other organisms like plants and fungi, it is different. I'm not really going to be talking about it in this course, but if you go on to take botany, you definitely talk about it there. And it includes different stages as well. So we do see different stages for other organisms. Questions here? Okay. I do want to also note that asexual reproduction does occur, of course. Now, this is reproduction where offspring are produced without the union of gametes, so only one parent is needed. If we think about advantages of this, there definitely are some. It takes a lot of energy, it takes a lot of investment, time, and sometimes for lots of organisms, it can actually be dangerous to engage in like mating practices. Sometimes with different animals, the male might actually be eaten. Sometimes male-to-male combat over mates can result in death. So it's not that sexual reproduction is not without cost. There's also a very high cost for forming like a placenta. If we think about placental mammals, for example, there's a lot of energy investment needed in that strategy. The benefits are there's a lot of raw material for evolutionary forces, and it's better. A lot of times it can be better or advantageous evolutionarily, but there are definitely advantages to asexual reproduction. Now, when this occurs, typically offspring are genetically identical to the parent. There are some variations. Mutations can still occur, which would be like a change in the genes. Sometimes that could be deleterious or bad or helpful. We also know with things like bacteria that reproduce asexually. I mentioned this quite a while ago, but they do have something called sex pili, which is basically like some type of bacteria can actually exchange genes, which is kind of cool. They don't all do it and they don't all have that feature, but some can. So this, again, does not result in genetic variation in a population. And what I'm showing here is an organism that is asexually reproducing. This is a hydra. So a hydra is a eukaryotic organism, and it belongs to the cnidarian family, which are basically like organisms with stinging cells. So other things that are in this group are like jellyfish, but hydra are like tiny little freshwater dudes. So the bud is just a localized mass of dividing cells. And I have a question for you. What process are those cells using to divide? Thoughts? Mitosis, does anyone agree? Meiosis? Okay, so let's break it. Let's think back. What does mitosis do? What is it helpful for? When we were talking about mitosis, like why does that matter? What's its value? So replication of what? Of chromosomes. So chromosomes replicate before mitosis, absolutely. But basically, we start with the parent cell, right? And then the whole point of mitosis is that we end with two identical daughter cells, right? It's just basically copying the cell, right? So mitosis is valuable for growth, development, reproduction occasionally for like asexual organisms. So asexual organisms can actually use mitosis to grow a bud, for example. So this hydra is growing this bud via mitosis. Now, some of you all were saying maybe they're using meiosis. Remember, meiosis produces gametes. So asexually reproducing organisms are not going to be growing buds or anything like that using meiosis. Does that make sense to everybody? No, they don't, because asexual reproduction basically just means you're able to copy yourself, and the production of gametes is for sexual reproduction, so you don't need to bother with that. Save the time, save the energy. Now, I do have a picture of redwood trees. That's what this is, by the way. It's kind of unclear, but they're actually also able to reproduce asexually, which is kind of cool. You might have heard, or at least like maybe have heard it in passing. I think it's been in a couple of news cycles. Trees are like very connected to each other, and a lot of times there can be trees are stronger when they're in forest groups, and a lot of times it can be related to this idea. Trees are able to reproduce asexually and form like huge, big underground kind of families, which is really cool. So getting back to the idea of meiosis, I put it in bold in like giant letters because this is super important. Meiosis is based on the interactions and distributions of homologous chromosome pairs. So homologous chromosomes, what are those? I'm just going to put this up here as a reminder for you. Homologous chromosomes are chromosomes that are the same length. They have the same centromere position, so the same little like waist point, if you kind of think about it, and have the same genes in the same order. Okay. However, homologous chromosomes are not identical because sometimes we see variations in the type of gene or in the form of the gene, which would be the allele. Now, a good example of this, I have two little orange arrows on the screen here. So we have these two little orange arrows. They're pointing to this thick band. Now on this figure, these bands are representing different genes, and they're at specific loci or locations. So we have these genes here, and let's say that this gene is pointing at eye color, okay? So maybe eye color on this chromosome, the allele, maybe this one's brown, maybe this allele is green. That's what I mean. Like it's the same gene, right? So it's coding for like the protein color in your eye, but we can see differences in the variation of that gene, in like what that phenotype is. Does that make sense? Okay, in reality, eye color is a lot more convoluted. It's more than just one gene in one location, but it's a simplified example. So this word homologous chromosomes is really important to make sure you're kind of studying this, remembering this idea. So same length, same genes, same location, but we get one copy from mom and one copy from dad when we're talking about sexual reproduction. Sound good? In this figure, I don't know why they use these two very similar looking colors. If you're colorblind, I think this would be a really challenging figure to decipher, but purple is represented, or whatever, like light purple fuchsia, whatever this color is representing the maternal copy, and this darker is the paternal copy. Questions here? Okay. So kind of just really reinforcing this idea, in diploid cells, in diploid organisms, the two representatives of each chromosome are called a homologous pair. So if you remember, I've shown you a human karyotype before. Remember, this is a visualization of all of the chromosomes that an organism has, and they take this when it is undergoing mitosis because that's when our chromosomes are maximally condensed. That's why we can visualize them in a karyotype. So we can't just take a cell in interphase and look at the chromosomes. They wouldn't be condensed enough for this visualization to occur. We have to take these when they're undergoing mitosis, which is kind of cool. Now, I like this figure because it's showing some really key pieces here. Our different genes are basically represented by color in these homologous chromosomes. Now, what's kind of confusing, and I recognize this, and we're going to keep working with this, but here we have a pair of homologous chromosomes. So within this pair of homologous chromosomes, we have 4 chromatids, right? Because remember, the chromatid is like one little line. So we have 4 little lines here, and those represent our chromatids. And then we have 2 duplicated chromosomes. We have 2 homologous pairs. So this is kind of trippy. This can make your brain feel a little funky. I recognize that. We're just going to keep kind of like working through that. As I was mentioning, homologous chromosome, same gene, same order. We already talked about this. I didn't have the second bullet on the last slide. The versions of the genes may differ. That's the allele. So the alleles might differ. I was already talking about that, maternal and paternal origin. Now, this is something really important. Each individual has a unique combination of alleles in the 2 chromosomes of each homologous pair. Yeah. Yeah, great question. So remember, gene refers to what it's going to code for. Genes code for proteins. And so, for example, maybe the protein is like taster protein on your tongue. And let's say there's 2 versions of that gene. Maybe you're a taster or non-taster. And those would be alleles, right? So the taster gene is like what the protein is going to look like. And the allele would be the variation of that gene. Does that make sense? So same kind of deal when I was talking about like eye color. We could also do like hair texture, for example. Like maybe there's the protein for hair texture and it's at a specific location on a chromosome. And maybe one allele is like curly and one allele is straight. And then the offspring might end up with some combination of those alleles. Maybe the offspring has wavy hair, maybe the offspring has straight hair, whatever. Say what? This picture here.
Which one of those chromatids look like the same?
Yeah, so each one of these chromatids is going to have the same genes in the same order, but they could have different alleles. Yeah, exactly. So they code for the same like broad structure, the same purpose, but maybe the specific phenotype is different. Another really classic example, and we'll talk about this a little bit more later, is like with flower color. For example, if you're looking at the flower color of a pea plant, the gene might be flower color, and the allele would be like purple versus white. So alleles are versions of a gene. There's something else popping up on here. Okay, so next class we'll talk about this little circle I just bopped up on here, but these are X and Y chromosomes, so more fun awaits. I do have office hours if you need after class, so feel free to stop by.
Quick question.
Sure, yeah, what's up?
Really bad about printing my labs in.
Uh-oh.